Hello, my name's David Paterson. I'm a Professor of Infectious Diseases at the University of Queensland in Brisbane, Australia. And I'm here with Professor Chip Chambers from UCSF in the United States. And we're going to talk about MRSA infections and also the news today about a new class of antibiotics discovered from soil. Just as a little bit of background, I'd like to ask you, Professor Chambers, really MRSA, what exactly does that mean and what's its significance for human health?
MRSA stands for methicillin-resistant Staphylococcus aureus. Obviously, MRSA is a lot easier to say. Methicillin is an older drug, no longer even available, but it was one of the first drugs developed for the initial problem with resistance in Staph aureus to penicillin. So penicillin, which became widely used in the 40s and 50s, was a highly effective drug. But because staph developed an enzyme that could destroy the drug, it fell out of use. And so methicillin was developed to overcome that problem. Methicillin-resistant Staph aureus is in fact resistant not just to methicillin, but to the whole class of antibiotics commonly used, called beta-lactam antibiotics, to treat staph infections. It's the most effective, the least toxic, vastly preferred drug class, and methicillin-resistant Staph aureus are not susceptible, with a few exceptions, to this class of drugs.
Excellent. So the research that's reported on from Rockefeller University describes the discovery of a new antibiotic class from soil. Now, we've got some history of discovering anti-MRSA drugs from soil. Is that right? Can you tell us about what we typically use or have used for the last 3 decades for MRSA infections?
A number of antibiotics have been identified in organisms that grow in the soil or closely related ones. So, for example, for MRSA infections, vancomycin has been a drug that's commonly used, and it is one of the drugs of choice. Daptomycin, also derived from an organism, a bacterial organism, has been used. The natural products really have been the backbone of drug development for treating all bacterial infections, including staph infections. Most are not synthetic. There are some exceptions, but in general, natural products have been a very important source of, and soil organisms also historically important source of antibiotics.
Mm-hmm. And why do we need new antibiotics for MRSA, but also for other bacterial infections? Why can't we just go on with what we've got at the moment?
Yeah. So the antibiotics that were discovered early for a reason. They're the proverbial low-hanging fruit. And drug development programs targeted drugs that were relatively easy to discover and that were relatively non-toxic. The easiest and the most effective were found the earliest. What has happened is, over the years, bacteria have developed mechanisms of resistance due to their constantly being exposed to antibiotics, such that the older antibiotics now are no longer effective because the bacteria have acquired genes and mechanisms that inactivate the drug or provide some other mechanism that allow it to avoid being killed by that antibiotic.
Now, we're not seeing a lot of resistance to vancomycin, but there's concerns about vancomycin's effectiveness. Is that right, or is that an overblown concern?
No, I think that, again, the penicillins and the beta-lactam class, like I mentioned, like methicillin, you would vastly prefer using those drugs. Number one, they're relatively well tolerated. But number two is they're more effective. So you're then in a situation with MRSA having to use a drug that is fairly well tolerated, but if only it were like a penicillin. It does not have the intrinsic activity that a penicillin does. Of course, you can't use a penicillin for MRSA, so you have to use vancomycin. But it doesn't work quite as well as a penicillin would if it were a susceptible organism.
Right. So it's very interesting, this new class, the malacidins, that rather than being discovered actually from the soil bacteria that you would grow in the laboratory, the investigators looked at unidentifiable organisms in the soil and were able to look at their DNA sequences and then derive what might actually be bacteria that have anti-MRSA properties. But it's early stages yet in the development of this antibiotic. I mean, what are the normal stages you'd have to go through from discovery, which is where we are now, and I guess early proof of principle that it works against MRSA, to it actually being used in our hospitals?
Right. So there have been many, many compounds that have been discovered that inhibit bacterial growth or kill other bacteria. But there are other steps along the way. The most immediate one is making sure that it doesn't have toxicity that would preclude its use in humans. And these might not all be in this particular order, but at some point you have to rule out that the drug is toxic and figure that out. Secondly, when administered, it has to have the right properties of how it's handled in the body. So for example, it might be a great antibiotic, but when administered to a human, it is bound up by protein and not available in circulation to inhibit bacteria.
Right.
So it's not chemically useful when provided to a human, or it's destroyed by some mechanism that the human host might have. So it has to have the right pharmacology. It cannot be toxic. That's rule number one for an antibiotic. It has to be incredibly well tolerated. Then, once you have figured that out, it has to be effective. So safe and effective. And the efficacy part is generally determined in 3 phases. The first 2 phases are more about the pharmacology that I talked about and the toxicity. The second phase incorporates something about how well it works, but then you have to conduct a relatively large trial to demonstrate that it's as effective or similarly effective as available drugs. And that step takes quite a while.
So what sort of time period are we looking at? You know, these researchers at Rockefeller have made this discovery, and it's a neat way of discovering an antibiotic, there's no doubt about that. But if everything went right with the toxicity and the pharmacology and the clinical trials, when would a drug like this actually be available to patients?
Best case scenario, 7 to 10 years, I
Wow.
think.
So 2025 plus. So a lot of hurdles. So, you know, I think we agree that Staphylococcus infections are common. They're killers. This news is great, but how would you sort of summarize the impact of this news in the whole scheme of treating antibiotic-resistant infections?
Of course, I don't want to discount the discovery of any antibiotic, but number one, I would say that the pipeline has been pretty good for developing drugs to treat Staph aureus infections. It wasn't so good 10 years ago, but it's quite good now. And the critical need is in gram-negative infections, and this particular antibiotic does not work against those types of organisms.
So this is the sort of germs causing infections from urinary tract infections or intra-abdominal infections, some types of pneumonia.
Right, and the most challenging ones currently for their problem of drug resistance. So I wish it were a gram-negative drug,
Right.
even though I'm a staph guy, because that's where the need is. Not that having an effective staph agent is a bad thing.
Yeah.
But so I wish that it had a little bit more utility against the gram-negatives, but we'll take it as a staph drug. But I would say don't hold your breath.
Mm-hmm.
It will take a while to go through each of the stages. Where drugs fail along the way is the compound that you discover may not be the antibiotic you ultimately develop. It may require chemical modifications to meet those other criteria that I mentioned. So that could take 3 or 4 years. There has to be testing to make sure it's safe, and then you have to test it against another antibiotic. So if it makes it past the first stage of safety in humans, then I think it would be interesting to think about at that point. Clinical trials in after then will be 3 or 4 years, I would guess.
Mm-hmm. So perhaps just to sum up, in a way, what might be most interesting about this news of this new antibiotic class is really the way it was discovered. More so in a way than that this is going to solve all of our antibiotic resistance issues from this particular class. Hopefully it's going to bring forward other classes of antibiotics active against other types of bacteria.
Yeah, this would be one of the first examples of the hope of knowing the genetic content of a bacteria to inform whether it would be useful in making antibiotics. It's usually done the other way. That is, use screening to see if it's active against bacteria. Here, it's a genetic approach that actually predicts, hey, this might be a good antibiotic. So if that bears fruit, it will suggest maybe a pathway for other antibiotics as well.
Excellent. Well, thank you for listening, and we'll be back with more antibiotic news as it becomes available. Thank you.